Hello, and welcome back to Teach Me in 10. In this episode, we're exploring a targeted gene expression analysis tool and its application for expediting translational validation and screening studies. Joining us is Michael Bailey, Manager of the Customer Experience Lab and nCounter Services for Brooker Corporation. Michael will demonstrate how the nCounter analysis system delivers high-throughput, targeted gene expression insights from bulk sample sources with outstanding accuracy. Already used by thousands of institutions globally, we'll hear how nCounter can produce robust results, even with challenging sample types. Over to you, Michael.
Hello, everyone. My name is Michael Bailey. I'm the manager of the Seattle Customer Experience Lab and our nCounter Services team here at Bruker Spatial Biology. Thank you for giving me the opportunity to introduce this fantastic fit-for-purpose technology. I hope by the end of this Teach Me in 10 episode that you'll have a better understanding of how this easy-to-use tool can help you further your own gene expression insights. I'd like to start by formally introducing you to the nCounter Pro analysis system, our third generation bulk gene expression platform, built specifically for reliability, reproducibility, and ease of use with your translational research. For those of us who may have some experience with this technology, or within the field of gene expression in general, you're likely familiar with nCounter's reputation for delivering high throughput, targeted gene expression insights from bulk sample sources with unparalleled accuracy. The platform is used by thousands of institutes worldwide and has been cited in nearly 8,000 peer-reviewed publications. We have dozens of ready-to-ship panels available, with the ability for custom targets to be added to each, or the option to build fully custom panels from scratch. But for many of us, this is our first time learning about nCounter. So let's start with a high-level overview. Let's call it an nCounter 101. We will begin by looking at the underlying chemistry, and then I'll cover our simple workflow, a bit more on our panels, an overview on common applications, and finally, a quick look at what's new in nCounter. In the simplest terms, nCounter is an analyte counting technology. While the majority of our panels are designed for mRNA detection, we also have chemistries that allow for the analysis of microRNA, DNA, and at the end of the presentation, I'll introduce our newest assay type, which we're all very excited about here at Bruker. A few key points on nCounter chemistry is that depending on panel, it's up to 800 plex per sample, with each of those up to 800 analytes capable of being measured tens to hundreds of thousands of times depending on biology. As you can likely imagine, being able to generate 5 linear logs of dynamic range for up to 800 different targets without need for any enzymatic amplification or cDNA conversion is a very powerful tool. And a wide variety of species and tissue types can be analyzed on nCounter. We like to say that if it has RNA, nCounter will work. We have built panels for mouse, human, fish, plants, canine, bovine, and many, many more. Our highly experienced application scientists and bioinformaticians can assist you in finding or constructing a nearly endless variety of designs to fit your needs. In addition, a wide variety of starting materials are acceptable. Importantly, nCounter is best in class with formalin-fixed paraffin-embedded samples. These are generally subject to extensive RNA fragmentation, making them challenging for many technologies. But nCounter's robust probe design excels in these conditions. And this list is not exhaustive. There are other sample types as well, including plasma, cerebrospinal fluid, and more. We're always interested and excited to work with novel or challenging sample types, so please reach out if you have questions. Let's quickly look at our probe design and chemistry. nCounter uses a 2-probe system, with each probe approximately 50 bases in size. These pairs anneal directly adjacent to each other and complementarily to your target. The small size of the probe system requires only 100 bases of intact sequence, hence why it works so well with fragmented RNA. The resulting probe target complexes are then ready for purification, imaging, then decoding and data generation, all fully automated on the nCounter Pro system. Unlike other gene expression platforms, nCounter has just 2 key steps for a total of 25 minutes of hands-on time. The first includes the direct hybridization step. This is conducted at the bench where the probe pairs are hybridized with your samples. After the overnight step is complete, this is an overnight hybridization process. On day 2, samples are then purified and loaded on the imaging cartridge by your prep station, followed by imaging, decoding, and raw data generation on the digital analyzer. A common scenario might be starting your project on a Monday and having data available to start your analysis that very next day on Tuesday. The data files are small in size, making them easy to work with and simple to understand. The digital output of counts per transcript can be analyzed using our free nSolver data analysis software, through our third-party partner Rosalind, or by accessing a wide variety of open source tools and code to conduct your own fully custom analyses. Another major aspect of the nCounter platform that makes it so easy to use is the portfolio of customizable thematic panels addressing all major research areas. Here we have listed the 25 panels we have in stock and ready to ship. They cover a wide array of research fields, and all of their content, including gene and pathway lists, design concepts, and any applicable papers or focus webinars are all available for free access and download on our website. And although entirely customized panels are available and popular, most users do prefer to start with pre-built panels, and I'll show you why next. For decades, the scientific community has been using a variety of discovery tools and sharing this data in the public domain. Oncology and immunology lead the way with available data, but many other research areas such as neuroscience and cardiovascular disease have amassed knowledge at the transcript level. This data serves as the starting point for nCounter panels. In an nCounter panel, we say it is not important to have data on every gene, but to assemble the knowledge that exists to create panels that can specifically address questions relevant to any given research area. We call these biological themes, and they are built using a data-driven approach along with real-world scientific input, and are not just a product you use on the nCounter platform, but a resource for your work that allows you to cut through the noise and get directly to actionable answers. But does the data correlate with what you might get from looking at other types of data? Let's take a look. In a cross-platform oncology clinical trial, scientists at the University of Sydney say nanostring nCounter pan-cancer IO360 can generate a similar immune profile to that generated by the whole transcriptome sequencing platform in advanced melanoma patients. In another large multi-center immunology project, this led by the Institut Pasteur in Paris, the project lead, Dara Duffy, shared with us that nanostring's gene expression panels feature key immune response in genes important to our study. We don't need to waste time focusing on genes that won't be induced, and it lends confidence to our statistical analyses. And in this third example, infectious disease scientists from Wake Forest University conducted a comparative analysis of RNA-seq and nCounter in lung organoids and stated that the concordance observed supports the complementary use of RNA-seq and nCounter. In each case, we see results that demonstrate strong correlation of immune profiles, pathway dysregulation, and gene signatures, all confirming that data generated using nCounter will yield the same biological conclusions as looking at tens of thousands of genes from whole transcriptome sequencing, but with much less technical effort and variability. Let's take a bit closer look at some of those key pathways and applications. Shown here is what we refer to as our menu of fit-for-purpose applications. 6 different applications commonly used throughout translational research, where having targeted insights from your data may not only be easier and faster, but it will help you get to the answers you need to know. Let's take a quick look at a few of these. We'll start with immune response. Here the question is often what type of immune dysregulation is taking place, and what are the mechanisms and processes affected. Immunology is rarely about discovering new genes, but more focused on an understanding of the overall immune response. And so building a panel to capture these major processes was key. Another similar example is when studying disease mechanisms by looking at pathway-based biology. Panels for oncology, for example, Focus on these well-characterized pathways and give special attention to representative genes. As is, the platform makes the perfect companion either upstream or downstream of your experiments, allowing for a connection between spatial gene expression and global gene expression, and all with a technology that is easy to use, highly adaptable for species and sample types, and requires no advanced data analysis skills to succeed with. But there is still plenty of room to grow within this technology. So before we conclude this Teach Me in 10, I want to quickly introduce you to the future of nCounter with our brand new multi-omics capability. This assay generates both RNA and protein expression data from a single sample through one convenient workflow. As of May 2025, we are in the late stages of development. We already have a pre-commercial early access panel available through our technology access program and are working on a wide variety of assay types that we hope to have available more widely later in the year. As you can see, the nCounter platform is very well supported, is here to stay, and will continue to evolve with new panels and new capabilities as a true multi-omics platform. I want to thank you again for your time and interest in the Bruker Spatial Biology nCounter analysis system. With its ease of use, robust performance, abundance of panels and customization, simple and straightforward data analysis, and with the exciting new multi-omics capabilities, now is the time to bring your gene expression work to the next level with nCounter.
This episode was brought to you by Bruker Spatial Biology, a provider of industry-leading spatial molecular biology solutions, consisting of instruments, assays, software, and services to empower researchers globally. By combining advanced technologies from nanostring and Canopy Biosciences, Bruker Spatial Biology has created an expansive ecosystem of the most innovative discovery and translational research solutions. To learn more, you can head to their website at brukerspatialbiology.com. Thank you for joining us for another episode of Teach Me in 10. If you enjoyed this episode, be sure to like and subscribe to catch all our latest releases.